Now, to begin the first step of meiosis, we must introduce some vocabulary. Now, this vocabulary that comes with prophase, or this vocabulary will be ever evolving as we continue through the stages, but in prophase we need to know the cell, which is represented by this red circle, the nucleus, which is represented by this brown circle, the chromosomes, which are going to be represented by these shapes here, which are made up of two sister chromatids, which are represented by the two pieces of paper that make up this formation, and the centromere, which binds these two chromatids together to form a chromosome. Now, these chromosomes carry pieces of genetic information used for to make the daughter cells have, well, genetic information so that they can be useful for sexually reproducing organisms. Now that you know this vocabulary, we will be able to continue with what happens during prophase. The genetic information is duplicated to form this. And not only this, but after it is duplicated, crossing over occurs, which means that these homologous pairs of chromosomes, otherwise known as two chromosomes that code for similar traits, are going to cross over. So this chromatid right here might take some of this chromatid's information, and this chromatid right here might take some of this chromatid's information because they're all jumbled up. Now, after this crossing over occurs, which it only serves to increase the genetic diversity of the four daughter cells, we can move on to metaphase. In metaphase, we see the disappearance of the nucleus. Goodbye, nucleus. Along with the formation of a synapse. Now, this synapse is simply a term for the structure that forms when the homologs, or the homologous chromosomes, form a structure here along the center of the cell. Now, during this formation, independent assortment can occur, which means this isn't the only pattern that the homologs can form in. It could form in this pattern, this pattern, this pattern, or what have you. This simply serves to further increase the genetic diversity of the daughter cells. When these spindle fibers appear right here, they attach to the centromere of the chromosomes, each of the chromosomes, and they are powered by a kinesin, which basically provides the energy that is needed to, once these meiotic spindle fibers attach, pull these chromosomes away from each other. And that is the purpose of the kinesins and the spindle fibers, is to pull them away from each other. Now, this pulling away from each other happens during anaphase. In metaphase, they line up, and in anaphase, they are pulled away from each other, off in different directions. This leads us into telophase. Now, in telophase 1, you see the cell, the parent cell, being split up into two daughter cells, along with the reappearance of the nucleus around each pair of chromosomes. What this serves to do is it serves to prepare until these two cells finally separate and become their own two daughter cells, each with a pair of two chromosomes and two chromatids within those chromosomes. And that is going to wrap it up for telophase 1, and consequentially the entirety of the meiosis 1 series. Now we will begin our journey into prophase 2. Now, in prophase 2, you're going to see the same ideas as you saw in prophase 1, however happening to two chromosomes instead of pairs of homologs. Now, I will be demonstrating it in one cell for clarity. However, it should be noted that this process takes place in each of the daughter cells. Now, in prophase 2, these cells have each one chromosome from each homolog pair. As you see here, We have one red and one blue chromosome indicating different homolog pairs. Skipping right ahead to metaphase 2, you see the redisappearance of the nucleus and the reappearance of this formation. The chromosomes line up amongst the middle of the cell. Now again, we see the reappearance of these meiotic spindle fibers, which are again going to attach to the centromere of a chromosome, which in turn will result in this chromosome splitting. However, since this is metaphase 2 and not anaphase 2, these chromosomes will not split just yet, as we will explain how this splitting differs from metaphase 1. Now, as you see, these meiotic spindle fibers are being set up in exactly the same way, attaching to the centromere and being powered by this kinesis, just being ready to be torn apart. However, you will also notice that no chromosome was torn apart in metaphase 1, or anaphase 1 I should say, because there were a pair. It was a pair of homologs that were being torn apart. Now, the difference between metaphase and anaphase 1 from metaphase and anaphase 2 is that only one chromosome, 2 chromosomes total, are being lined up across the center plate, not forming a synapsis. And these meiotic spindle fibers are going to pull apart 2 chromosomes, resulting in the disappearance of the centromere. When they split and go their separate ways, the sister chromatids each go in their own direction. Now this will happen in the second chromosome as well. When they are being lined up and ready to be split, this is known as metaphase 2. When they split and the meiotic spindles pull apart and you end up with the end formation that you see here, this is known as anaphase 2. Notice how the nucleus is still not reappeared. Now this process, as mentioned before, only happens in one cell in our demonstration, but it should be noted that this will happen the exact same way in the other daughter cell. In telophase 2, the spindle fibers again disappear, similar to telophase 1, and the nucleus again reappears, as you'll see, similar to telophase 1, except by the end of telophase 2, we will have now 4 total daughter cells at the end of meiosis, each with one chromatid coming from the homolog that we started with at the beginning of meiosis 1. This is the end result, and although it seems that these 4 daughter cells are genetically identical because of the examples, because of crossing over occurring, we will actually see that these are all genetically different. That is to say, no 2 chromatids will be genetically identical. That marks the end of meiosis 2 and consequently the end of meiosis. Thank you for watching.